My name is Martin. I'll start again. So good morning, good afternoon, everyone. My name is Martin Hurst, and I'm a professor in the Michael Smith Laboratories at the University of British Columbia, and I will be your host for today's seminar. Before we begin, I would like to take a moment to acknowledge that Canada is on a journey recognizing the gap between the democratic ideals that it was founded on and the realities we've experienced over the past 200 years. Although we gather today on a virtual platform, I would like to honor the Indigenous peoples whose lands we are on, wherever they are located. This reminds us of our shared relationships to the land and to one another as we strive together towards realizing the ideals for all. So a reminder that today's seminar is virtual and a virtual webinar, and questions can be posed through the Q&A box. So please use the Q&A box and not the chat box for any questions. And the panelists will be monitoring those question boxes as they come in. So now it is my distinct pleasure to introduce our first speaker for today, Signe MacLennan. She's a Ph.D. candidate in the Marra lab, whose thesis research surrounds investigating the interplay between DNA methylation and structural variation in virally driven cancers. And today she will give us a talk entitled The Structural and Regulatory Architecture of Extrachromosomal DNAs in Cervical Cancer. Signe?
Thank you for the introduction. I'll just share my screen here. You see that?
I do, yeah, looks great.
So thank you for the introduction. Yeah, I'm Signe and I'm a PhD candidate in the Marra lab. And today my talk is titled The Structural and Regulatory Architecture of Extrachromosomal DNAs in Cervical Cancer. So despite available human papillomavirus or HPV vaccines, cervical cancer remains the fourth most common and deadly cancer affecting females globally. And in large regions of Africa, cervical cancer is the deadliest cancer affecting females. And extrachromosomal DNAs, or ecDNAs, are a large circular species of DNA that drive a subset of malignancy via multiple mechanisms. So they're best described in the literature as what's called oncogene amplification. So ecDNAs often contain oncogenes and/or their associated enhancers, and then become selected for to be at very high copy number in cancer cells, thus driving high oncogene expression. Similarly, ecDNAs are associated with treatment resistance, for example, by amplifying drug resistance genes. And ecDNAs also lack centromeres, so are randomly segregated to daughter cells during mitosis, which can generate significant intratumoral heterogeneity in terms of the number of ecDNAs per cell. And ecDNAs have also been reported to reintegrate back into the chromosomal DNA, wherein they've been reported to disrupt tumor suppressor genes or potentially further increase genomic instability, as ecDNAs are themselves a product of increased genomic instability. And what's interesting about ecDNAs in HPV-driven cancers, including cervical cancer, is that ecDNAs can sometimes contain parts of the HPV genome. So these so-called HPV human hybrid ecDNAs are formed via the process of HPV integration, wherein the HPV virus incorporates its genome into that of the host. But beyond this, the functions and regulatory properties of HPV human hybrid ecDNAs are not well understood. Furthermore, we know from the literature that approximately a quarter of primary cervical cancer tumor samples contain evidence of ecDNAs, but very little is known about ecDNAs in the advanced stage context. So thus, we hypothesize that the structural and regulatory landscape of ecDNAs is dysregulated in cervical cancer, and that the patterns of this dysregulation differ between HPV human hybrid and ecDNAs containing only human DNA, or human-only ecDNAs. And we had two main driving questions. So what is the structural and epigenetic landscape of ecDNAs in cervical cancer? And in terms of epigenetics, we looked both at methylation using long-read whole genome sequencing data and histone modification data from ChIP-seq data. And secondly, how do HPV human hybrid ecDNAs differ from human-only ecDNAs in terms of their structure, regulation, and predicted function? So our cohort consisted of 118 mainly advanced stage cervical cancer tumor samples from a patient cohort in Uganda that were previously profiled with both short-read whole genome sequencing and RNA-seq. We had 50 of these samples with available ChIP-seq data, and more recently, we profiled 47 of these samples using nanopore long-read whole genome sequencing. And so what did we find? We found that ecDNAs appear to be prevalent in cervical cancer. So we bioinformatically detected 50 putative ecDNAs spread across 30% of samples, with 13 of the 50 ecDNAs being predicted to be HPV human hybrid ecDNAs. We also observed that nearly 30% of ecDNA positive samples were predicted to contain multiple species of ecDNA, with up to 4 observed in one sample. We were also interested in looking at the expression of oncogenes found on ecDNAs, as this has been reported quite extensively in the literature. So here to interpret this graph, the ecDNA sample is in the red dot, and that sample contains an ecDNA bearing the corresponding oncogene, whereas the rest of the box plot is the rest of the cervical cancer cohort. And when compared to the rest of the cohort, oncogenes tended to be highly expressed in the sample predicted to contain an ecDNA bearing that oncogene, although this was not universal across all oncogenes. And this is consistent with previous literature on ecDNAs as mediators of oncogene amplification. We were also interested in what parts of HPV are contained on the HPV human hybrid ecDNAs. And we found that all of them contain the HPV oncogenes E6 and E7, here shown in yellow. So here the height of the red bar indicates the number of ecDNAs containing that region of HPV. So E6 degrades P53 and E7 results in the degradation of retinoblastoma protein. So both of these are important tumor suppressors in cancer. I'll also make a couple other notes here. So the HPV human hybrid ecDNAs infrequently contained an intact E2. E2 encodes a negative regulator of E6 and E7 and is commonly lost or disrupted in the process of HPV integration. And all hybrid ecDNAs contain the long control region, or LCR, which has important regulatory properties and contains the virus's origin of replication. Next, we compared kind of the genomic landscapes of HPV human hybrid and human-only ecDNAs. And we found that HPV human hybrid ecDNAs tended to be smaller than human-only ecDNAs and contained and had a tighter distribution of sizes. And likely related to their smaller size, they contained fewer human genes than human-only ecDNAs. Next, getting into more of the epigenetic side of our study, we looked at available ChIP-seq data to look at enhancers on ecDNAs. So there's a caveat here that we can't isolate the ChIP-seq signal specifically to the ecDNA, but if we look in the ecDNA locus, the ChIP-seq signal should be enriched for the ecDNA as they are present at high copy number. So overall, we found that although human-only HPV human hybrid ecDNAs tended to contain similar numbers of annotated enhancer loci, only HPV human hybrid ecDNAs contained significantly elevated H3K27 acetylation activity in these loci compared to ecDNA negative samples. And this was not significant for human-only ecDNAs, perhaps indicating that HPV human hybrid ecDNAs are more likely to be serving as enhancer vehicles than human-only ecDNAs. We were also very interested in looking at the methylation status of ecDNAs in our cohort, as this is an underexplored area of the literature. So we did this by first utilizing what are called allelic differentially methylated regions, or ADMRs. So very simply, when alleles have different CpG methylation states within a particular genomic locus, we term that locus an allelic differentially methylated region based on some statistical significance. And we make a couple of assumptions. So ecDNAs, we assume arise from a single allele and are present at high copy number. And so we would expect that if ecDNAs have significantly different methylation states compared to the chromosomal DNA, we might expect them to be enriched in ADMRs compared to the rest of the genome. And we found actually that this was not the case. So ecDNAs are not enriched in ADMRs as compared to the rest of the genome. Although they do contain ADMRs, we find that they rarely overlap CPG islands. Less than half overlap genes, and most of these fall in intronic regions. However, we did uncover an interesting example of two ADMRs in an ecDNA locus that overlap the promoter of a gene called C7orf65, which encodes a link RNA. So here you can see haplotype 1 is relatively more hypomethylated compared to haplotype 2 within this gene. This haplotype 1 is predicted to be the ecDNA haplotype. And we looked also at the RNA expression data for this gene, and we found that this gene was expressed in about half the cervical cancer cohort, including this sample, but only expressed in 1 out of 19 normal cervix samples from GTEx, perhaps indicating it has some cancer-specific expression. So in summary, we compared the genomic and epigenomic landscapes of human-only and HPV human hybrid ecDNAs in a cohort of mainly advanced stage cervical cancer tumor samples. And we found evidence that human-only ecDNAs and HPV human hybrid ecDNAs are distinct. So human-only ecDNAs tended to be larger and contain more human genes, whereas HPV human hybrid ecDNAs invariably contained the HPV oncogenes E6 and E7 and tended to contain more active enhancers, whereas both ecDNA types contained few ADMRs, which rarely overlapped promoter regions. And so overall, we hypothesized that given our evidence, human-only ecDNAs are mainly serving as oncogene vehicles, whereas HPV human hybrid ecDNAs are mainly serving as E6, E7, and enhancer vehicles. So next, we're planning on validating a subset of the ecDNAs using a molecular approach called CRISPR-Catch. Using this approach, we may be also to specifically determine ecDNA methylation with less noise from the chromosomal DNA, as for now, we've been relying on haplotype phasing. And also, if you're attending the conference in Blue Mountains in October, please come say hi and check out my rapid talk and poster. That's in a couple weeks. And finally, if you want to learn more about Marra Lab HPV research, please check out some of our lab's previous publications. So this 2020 Nature Genetics paper, and currently a bioRxiv preprint, but soon to be published study led by Dr. Vanessa Porter in our lab that looks at the epigenomic and genomic features of HPV integration in cervical cancer. So with that, I'd like to thank everyone in the Marra Lab, especially my supervisor, Dr. Marra, as well as all our funders and the patients and their families, without which this research would not be possible. And I'm happy to take any questions.
Wonderful. Thank you, Signe, for a fascinating talk. While we wait for some questions to come in, and a reminder that these need to come in through the Q&A box, I will get us started here. So just a really basic and maybe selfish question to start with. How easy is it to detect these ecDNAs, and what input data do you need? So let's say is it short read, long read? Is this an established pipeline?
So yeah, that's actually a very good question, because ecDNAs they're quite large, so they're hard to detect. We've been relying on a short read tool called Amplicon Architect because we could run it across the whole cohort. But there are a number of limitations to using a tool like Amplicon Architect because it's based on short reads. So I've been validating the ones with available long read data using a tool called Fly, which is basically a de novo assembly-based tool, which also predicts if a contig is circular. In like cell lines, you can detect ecDNAs using methods like FISH. And now we're going to validate a subset of them, as I mentioned, using this more molecular approach where you called CRISPR-Catch, where you do need to know a priori how large the ecDNA is in the sample. And then you basically do a gel extraction of the band. And then you can do long read sequencing. So we can also determine methylation of the ecDNA that way. Does that kind of answer your question? Sorry, it's
No,
the
it
hard
does.
thing in the literature to detect ecDNAs just purely using short read sequencing data.
Got it. So it requires levels of validation, including potentially long read sequencing to confirm, although you're making predictions and building models from that. So that's helpful to know. Thank you. So there are a few questions that are coming in. So I think I'll start with the first question from Signe, which is, did the presence of HPV human hybrid ecDNAs correlate with the reduced RB1 and P53 expression?
I did look at this, I believe. We have to keep in mind that these are all HPV-driven cervical cancers. So I don't believe there was a correlation between the presence of an HPV human hybrid ecDNA and reduced expression, because the other samples in the cohort also, most of them have HPV integration. It just wouldn't be in the form of an ecDNA. So E6 and E7 are kind of required in terms of expression to drive cervical cancer. So we'd expect P53 and RB expression to be low in all of our samples.
Okay, thank you. So the next question is, what are some, and this is from Anthony, what are some of the reasons that might explain the differences in size between human-only and hybrid ecDNAs?
Yeah, so this is something we've thought about. So my current hypothesis is that the minimum cargo that an HPV human hybrid ecDNA needs is likely E6 and E7, maybe an enhancer. But E6 and E7 are not very large genes. So the whole HPV genome is only 8 kb. So this would, whereas human genes, like human oncogenes, can be much larger in size, and there's a lot more variability, of course, but we predict that the size difference is likely driven by the difference in cargo size.
Okay, thank you. And the next question also from Anthony is, what is the process of ecDNA generation and how prevalent are they? So is this something that's unique to HPV, or you see this more generally, and how prevalent are these?
Yeah, so ecDNAs can be formed via multiple mechanisms. So one of which is chromothripsis, for example, or there is a model called the episome model, where it's a little bit simpler, where in ecDNA, there's just 2 double-stranded breaks, and then the intervening sequence recircularizes. The HPV human hybrid ecDNAs are obviously formed in the process of HPV integration and excision. But in terms of ecDNAs more generally across cancers, there was a 2020 Nature Genetics paper that looked at this, and they found that across malignancies in the TCGA with whole genome sequencing data, about 14% of cancer samples are predicted to contain ecDNAs, but that this varies widely by cancer type. So in something like glioblastoma, we see over 60% of samples contain evidence of ecDNAs. And also it's high in sarcomas as well. Whereas like a lot of the hematological malignancies don't have a very high frequency of ecDNAs.
Okay, thank you. And I think we have time for one more question, eating a little bit into Marco's time, but I know Marco is okay with that. So the question is, for human hybrid HPV ecDNAs, are these recognized by the CGAS or other sensors? In other words, other sensors that sense the presence of viral RNA. Is this known?
Sorry, can you repeat the question?
So the question is whether the human hybrid HPV ecDNAs are recognized by the CGAS sensors, which sense the presence of viral yeah.
That's a very good question. I don't believe there's any real literature on this. I mean, I could be mistaken, but that would be something definitely to look at. HPV human hybrid ecDNAs have been reported in the literature before, but I don't know if anyone's looked at CGAS. Yeah.
Wonderful. Thank you, Signe. So there are a number of other questions that have come in the QA box. We'll answer those, or Signe will answer those by typing, so that we can move forward with our next speaker. So it's my pleasure to introduce our next speaker, Dr. Marco Marra. So Dr. Marra is a professor in the Michael Smith Laboratories in the Department of Medical Genetics at the University of British Columbia. He's a Canada Research Chair in Genome Sciences. Marco has a long list of awards. I'll just name a few here. He's been appointed to the Order of British Columbia. He's a Fellow of the Royal Society of Canada and the Canadian Academic Health Sciences. And he's received honorary doctorate science degrees from Simon Fraser University and from the University of Calgary. He obtained his Ph.D. from Simon Fraser University and did his postdoctoral work in Washington University at St. Louis, contributing to the Human Genome Project. His current research focuses on genome science and its applications to human disease, particularly cancer, studying cancer genome biology, treatment resistance, and integrating genomic technology into cancer care. And with that, I would like to welcome Dr. Marra, who will speak to us today about his recent work in multi-omic characterization of pediatric AML. Marco?
Great. Well, thank you very much, Martin. And thank you, Signe. That was great. So that's me, and that's what I'm talking about today. My intention is really to provide a bit of a survey of some of the work we've done in pediatric AML, provide you with a bit of a sense as to some features of the disease, and then some plans to extend our studies into the single cell space in a perhaps more meaningful way than we have done previously. Before we get started, I feel I need to disclose that I do some things for the Terry Fox Research Institute. Here they are, and thank you all very much for supporting the run, which happened this last weekend. Much appreciated on behalf of patients and researchers and the communities everywhere. So this is a cancer talk, and Signe gave a cancer talk. And I always like to start my cancer talks with this slide, which summarizes basically two important papers that describe hallmarks or features of malignancy. And these are classics from Hanahan and Weinberg in two different publications, a decade apart in Cell. I'm sure we've all run across them. And the way that I think about this diagram is an illustration of cancer heterogeneity at various levels. And so we have this depiction of a mass in the center of the diagram, and not all the icons in the mass, which are meant to represent tissues and cells, they're not all the same. And we now understand, I think, largely through interrogations of genomics that the heterogeneity in that space is very, very high. We have a heterogeneity of processes that are dysregulated, and we have a heterogeneity of treatments that target this dysregulation. And so when we think about cancer, we have to accept, I think, that it is a disease defined by heterogeneity, and heterogeneity is quite likely its most powerful elusive feature in terms of treatments that could combat it. So cancers are heterogeneous. How do they affect the Canadian population? So this is a slide from the Canadian Cancer Society Statistics Handbook that we can all download and look at. And it basically shows the burden of new diagnoses per age group. And what you can see are the 0 to 14s on the left, and that's the space we're largely going to look at today. But the spectrum of malignancy that affects this age group is notably different from that of all others. And you can see that cancers that are major causes of mortality really are quite different. And so the leukemias, although they account for 35% of diagnoses, account for about 23% of deaths. AML is in that bin, pediatric AML. So about 25% of those diagnoses will be pediatric AML. And in terms of treatment efficacy and outcomes, approximately 80% of pediatric AML patients will achieve a complete response after induction chemotherapy, but a significant fraction go on to relapse. And when they do, the outcomes are dismal. And so it's these relapse cases and the processes that might separate disease that is prone to relapse from disease that might be curable, insofar as anybody is ever cured of their cancer. Those are the processes that are of interest to us. And so we very much value the opportunity to examine cohorts where we have a diagnostic sample available, where we understand what the treatment was, and where we have the opportunity to look at another sample from the same patient when they may relapse. And so our first opportunity to do that is shown on the left panel. These are extremely dense slides. I apologize. They can be hard to look at, but the message is I think, quite simple. And so on the left, this is work from quite a while ago from Dr. Amelia Lim's PhD thesis, in which Dr. Lim created a signature, or a model, if you like, that was able to anticipate outcomes in a pediatric AML cohort. And we did this looking at microRNAs predominantly. So more than 1,000 micro samples from which we collected microRNA profiles. There were some treatment refractory samples. These are ones that failed to respond outright to treatment, and then a limited number of relapse samples. And so this microRNA profiling was used to train a model. There's a little diagram over here on what the strategy was. And then we were able to show that in the discovery and validation cohorts, that model scores correlated with treatment outcomes. So if you had a high score, you did poorly with respect to event-free survival. And if you had a low score, you did better. So something about the gene expression space, particularly in the microRNA space, could be correlated to a treatment outcome, and this was significant to us. We then went on to work in both a mutational space now, in both pediatric and adult cancers. And so on the right side of the panel over here, we just highlight some differences that we see in pediatric AML versus adult AML. And so this top panel, mutations per megabase, this is a log scale. And you can see that pediatric AML has a relatively low mutation burden compared to adult AML. We can see that the pattern of copy number alterations is distinct, with the adults being in red and the pediatric cases being in blue. And when we look at the frequency of cytogenetic abnormalities, in cases we see that younger patients have much higher frequencies of these cytogenetic abnormalities than do older patients. And focusing on a few of the most highly mutated genes, at least in certain contexts, we see great differences. So here's KDM5A, KMT2A, and you can see that in the pediatric cases, the mutation frequencies are clearly illustrated. These are fusions, it says. That's what the pink thing means. And if you look over here at adults, KDM5A is not dominant and KMT2A is much reduced. And you can see overall a very different spectrum of alteration. So big differences between pediatric AML and adult AML. In other words, pediatric patients are not just little adults. It's a different disease. Okay. So another feature that we like to think about are fusions. And so we talked about cytogenetic abnormalities in the last slide, and a certain class of cytogenetic abnormality might be a fusion, if you like. And so when we look at fusions, we see that here they are, here are the mutations. When we look at fusions, we see a very high proportion of cases with fusions involving a gene called KMT2A. And KMT2AR means KMT2A rearranged. And so in this particular arrangement, something is glued on to KMT2A. And these rearrangements or general dysregulation of KMT2A function can be associated with different risk groups here on the right. So there's low risk, there's partial tandem duplications, and then there's high risk, but most of the KMT2As fall into this high risk category. I hope you can see my mouse. So there's a lot of them, and they tend not to do very well, I think is the point I'd like you to absorb here. So KMT2A rearrangements do not only involve one partner gene. There are many. Some say 80, some say 100. There are some dominant ones, which are more prevalent, but there are a lot. And certain of these KMT2A rearrangements are associated with different outcomes. And so Dr. Rachel Wong in our lab computed these data from a clinical trial and was able to show that these curves are different, and they seem to correlate with the partner that is glued on to KMT2A. This little diagram down here simply tries to illustrate the proportion of cases that have the distinct rearrangements. And so this, whatever color this might be, correlates with the MLLT3, whoops, apologies, and so on and so forth. And these can have different functions. Some are involved in DOT1L recruitment, some are involved in dysregulation of signaling molecules, elongation complex deficiencies, and so on and so forth. So a wide range of different alterations. But all of them seem to have something to do with KMT2A, which is a histone methyltransferase that acts on H3K4. And I will say at this point, because I think it's important to do so, that there's a whole different class of diseases that occur as a consequence of loss of function alterations, well, or in some cases, not so clearly loss of function alterations, as syndromes, especially in young people. And that's what the right-hand slide shows. So you can see the various marks affected, the genes, and in the center, the syndromes that they seem to cause. And so KMT2A is right down here, and you can see Wiedemann-Steiner syndrome. Another gene that we study a lot in the lab is KMT2D, which is associated with Kabuki, and there's KDM6A, remember the same complex as KMT2D, also causing this syndrome called Kabuki syndrome. So these are important genes. The proteins are important. They cause disease across a range of different phenotypes. So we then took the step of speculating that a great deal of bulk analysis had been done, and we had done a lot of it, without generating any particularly motivating insight around how primary and so that's at diagnosis and relapse disease might differ. And we, or we being me, didn't feel as though we had fully exhausted this space. And certainly I didn't feel that the insight gained was quite yet satisfying. And so we took a single cell approach to this most recently to do a preliminary experiment. And so the idea with the single cell analysis is that in the old smoothie analogy, which I'm sure many of you have seen, it's attributed to Aviv Regev, you have this is the whole genome sequencing bit where you homogenize all manner of different cells and you end up with this picture. And so you get a sense that, yep, it's pink and gooey. Whereas in our single cell space, we'll find the blueberries. And it was really the blueberries that we were after. We wanted to be able to say with single cell precision, what is happening to the cells in the primary disease and what is happening to the cells in relapse disease. And in the experiment I'm about to show you, we also had remission samples from patients, which we could use as some semblance of normal. So single cell seemed like a good idea to us. And then this business of serial samples I've already talked about. This is my very poor illustration of tumor cells and the niche. And here I illustrate a yellow type of cell, a yellow phenotype expanding, a green phenotype expanding, and the absence of other phenotypes. So I hope that illustration helps, although some tell me it makes things more confusing. But anyway, you can let me know. So we wanted to explore this in a proof of concept sort of way, and we had an opportunity to do so in the context of a clinical trial, AML 1031. And there's the title of the trial. You can learn more about it on clinicaltrials.gov. We had quite a number of primary remission and relapse samples available to us. There was good corollary information, whole genome sequencing, RNA-seq, and against that backdrop, we were able to create single cell libraries from I don't know what it was exactly, but anyway, there were 140 single cell libraries, dozens of samples across this space. About 700,000 cells were profiled, and we were able to marshal the data in such a way as to publish it in this cancer cell paper that was published, I believe, late in 2022. And Sander was a postdoc at the time in our lab, who is the first author on the paper. Just to highlight some of the things that we observed. So this cohort that we profiled was a very heterogeneous one and very small numbers of patients. So nothing I'm about to show you is particularly compelling, and that's something that we think we need to address in the next phase of our studies. But the point here was to simply ask whether we could generate single cell RNA data, whether we could generate single cell attack data, and if we could, did this preliminary survey see anything? And at this time, and maybe still, although it was fragmented and heterogeneous, was I think the largest pediatric single cell data set that I knew of, certainly the largest pediatric AML data set. And so there was some enthusiasm for this in the community. So there's the number of patients we had per disease type. We generally were working with bone marrows, marrow biopsies. So these are aspirates of the bone marrow in kids with cancer. And as I said, we generated single cell RNA and attack-seq data. And so this is the kind of thing that we were able to see as we were really getting our hands dirty with the single cell data. So this is a single patient. This is an RNA-seq data set. You can see in red here the cells from the diagnosis, and in green the remission. There's the green guys, and then in blue the relapse. So this is a UMAP projection, and I really don't want to get into a huge defense of whether UMAP is a good thing or not. It's the thing we used, and this is what it showed us. We were also able to integrate the single cell attack data and use it actually separately to generate these pictures. This is the same patient, but in a different data domain, and you still see separation between diagnosis and relapse samples. One of the things that we felt we needed to do was have a clear marker for mutant cells. So you generate all these profiles, but how do you know which cells are the mutant cells? It turns out to be non-trivial in this context. And so we leveraged the whole genome sequencing data, identifying somatic mutations from the whole genome sequencing data, and then looking at both the RNA-seq reads and the attack reads for overlap with these data. And what we were able, whenever we saw a mutation in the single cell space, we colored that cell red. So all of these are mutation bearing cells. And it's not a lot. I mean, it looks like a lot from the picture, but it's a few percent only. Remember, the mutational space of these cancers is very low. And so, but even so, this was able to reveal these mutant cells, these malignant cells. Okay, so we have malignant cells in hand. We can focus on the malignant cells, and we can ask what happens to these malignant cells across diagnostic and relapse samples. This is just an illustration of the kinds of things that we were able to see. So different diseases, different causes of pediatric AML are shown here for 3 ITDs and the MLL class. And so if you look under biopsy, you can see here's 2R and 2. So that's patient number 2, relapse, and patient number 2, diagnostic. And so you can see they separate. In some cases, they separate a lot. And so you get things like 6R and 6 in this UMAP projection. I know questions about biology here, but we shouldn't infer too much from the picture, but they do separate. And in other cases, it's not quite so much. And here's patient 8, for example. When you layer cell type annotations that you infer from a very complicated process, but largely related to gene expression, you cell type annotations onto this, you can start to see the relationship between relapse disease and the cell type that seems to be enriched in relapse disease. So if you look at the 2R2 example here, and you hop over to cell annotation, 2R looks like it's got a lot of these gold things, and 2, the diagnostic sample, has a lot of these blue things. You can think of these as the gold things as being primarily related to an HSPC or a primitive-like cell population, and you can think of the blue things as being more myeloid. And so when you follow that through with overlay of cells that you think are just the HSPCs, stem and progenitor-like cells, you start to get these pictures emerge. So here we go again. HSPCs are there in the relapse, but less so in the primary. And you can diagram this out across all the samples in that fusion class. And so at diagnosis, you get this proportion of HSPC-like cells, and in the relapse, it's greater. And you don't necessarily see this amplification of this cell type across different rearrangement partners or different genetic causes, different cytogenetic classes. So the FLT3 ITDs, they don't behave in exactly this way. So this seems to be special to this particular class. So I want to talk a little bit about how we then went about identifying some transcription factor binding site motifs. And so we used single cell attack-seq to do this. And the general paradigm was to create clusters of related cells, and then create what we refer to as synthetic bulk by combining the data from these clusters using the rationale that the clusters contain very highly related cells. And we could combine the data from these cells, combine the single cell attack data from these cells, and generate peaks. And so I'm sure, given this audience, we're all aware, so I won't really dwell on it, but these peaks that we were seeking to capture represent regions of open chromatin that are tagged in the attack procedure. So when we had these peaks, we then looked under them for enrichment of transcription factor binding sites. And that's what we see here. I think the left-hand side of things is simply an illustration of diagnosis and relapse. And we've seen that before, and an illustration of myeloid versus HSPC. We've seen that before. I've just spoken about that. This is the bit that I'd like to focus on. So with these peaks in hand from these synthetic bulk samples that we created artificially, we can ask for differential enrichment of peaks. In the diagnosis, that's red, versus the relapse, that's blue. And then when we look at the transcription factor binding sites under those regions of presumed open chromatin, we can see different transcription factor families. These are enriched in diagnosis, and these guys are enriched in relapse. And this can be marshaled to infer in D down here that the regulatory landscapes between primary and relapse disease are distinct. And that's all that this illustrates, are these distinct regulatory landscapes. So we thought that was pretty interesting, and wanted to think about ways that we could follow through with that, remembering that the most predominant shift in cell phenotype we saw was with the KMT2A rearrangement partners, which are also bad actors in terms of clinical outcomes, and also seem to play a very important role in regulating these transcription factor networks. So we want to drill down now on the KMT2A rearranged guys. And so Dr. Rachel Wong generated a few views for me to share with you today. And so the point here is that the KMT2A rearranged cancers, which are shown in purple, produce these lovely diaphanous structures on this UMAP projection. There are quite a few of them compared to the RUNX-driven cells and the CBFB and so on and so forth. Pattern is less distinct in the attack, but it's nonetheless there. This diagram on the left here just shows the samples. So the purple guys correspond to the purple guys here, and the green guys correspond to the green guys. That's probably straightforward. Another measure of diversity that Dr. Wong used here was the Shannon Diversity Index to show that the KMT2A rearranged cases have more diversity than the other types. If you look at the KMT2A cases only and you segregate out the cells by their partner, you get the following picture. And so AFF3 is a partner. There's the KMT2A rearranged AFF3. There's its expression. MLLT3 looks a bit more diverse. MLLT4 is up here. SEP6 here. So distinct gene expression profiles that correlate generally with these UMAP projections. If you look at how the gene expression patterns correlate with diagnostic versus relapse disease, Dr. Wong can compute a picture that looks like this. And here we're focusing only on the primitive-like cells, these guys here, the proportions of which expand in the relapse, that's shown there. And the point basically is that for each rearrangement partner, there seems to be very clear separation of gene expression pattern in the diagnosis. The same is true at relapse, but the gene expression patterns are different. And this just confirms in a slightly different way the types of results that Sander was able to achieve in the study that I've shared with you just a few slides ago. Dr. Wong then is able to use the attack data and for each rearrangement partner, ask what are the open chromatin landscapes in a similar way that we did in Sander's experiment. And so when KMT2D is stuck with MLLT3, what are the regions of open chromatin? And so here they are again, enriched in diagnosis and enriched in relapse. And so we can see some similarities in terms of, enriched in diagnosis. So we see some names here. We see some of those same names here. We see some different ones here. Again, some similarities and whoa, some different ones. And the same goes for the relapsed disease. So there's diversity here. There's heterogeneity here. These are driving potentially different downstream pathways. So heterogeneity is here, even though it's not necessarily driven by mutation. It may be constrained somehow by the nature of the rearrangement partner and the biology that it can drive. We don't know. But there does seem to be some rearrangement partner specified differences in the KMT2A rearranged space. All right, now Martin has been waiting for this moment because I'm about to share with you a custom created cartoon. And Martin has seen many of these over the years, and all of them are equally confusing, and I will show one that lives up to that history. So let's assume that we have KMT2A rearrangement partners. There they are, different colors, different ones. R1 through R5 just means they're different. So we know that these things sit on top of regulatory cascades. And so they partner with genes, they have action at various regions in the genome, and they turn on other things. So each one of them is associated with a regulatory cascade. There may be some overlap between cascades. There probably are, but there's going to be some unique things. And because these regulatory cascades often involve transcription factors, they too will activate downstream patterns of gene expression. And eventually you get yourself into a situation when you try and think about what might be going on, where you decide that this is probably the most appropriate picture, a hairball of interactions. And the question is, well, how do we deduce where our targets might be and where the opportunities for future development of therapies might be, which is a major problem in this disease. There is an alternative, and the alternative is that, well, yes, this is all true, but somehow it all funnels down into some pathway that leads magically to a single treatment. I don't know, but I suspect that this is not a very realistic scenario. I think much more work needs to be done to try and deduce whether or not we're dealing with a situation where real precision medicine will be required to match rearrangement partner subtypes with treatments, or whether we can realistically expect in this class of pediatric AML to have a thing. I will point out that the clinical trial that we were from before, the Sander example and the Rachel examples, that was from a failed clinical trial testing a drug called bortezomib, which is a proteasome inhibitor, which has shown activity in adult AML and multiple myeloma, but completely failed in the pediatric cancer context. But it's, there was a good rationale for it, didn't work. Okay, so what do I think we can do in the Marra lab going forward? Well, remembering that we had only very, very small numbers of samples and we can't deduce anything general about any of these classes, we think that an obvious thing is to sample more deeply. And so we've been building a cohort with our colleagues Sohail Mashinchi and Rhonda Reese at the Hutch, Fred Hutchinson Cancer Research Center in Seattle. And we're busy enumerating what we have available to us, and we fully intend to embark on another round of single-cell multi-omics there. We're going to look at primary versus relapse disease when we can, and we're going to generate whole genome sequencing for all of these, and probably on a methylation-sensitive platform. And if I could have single-cell methylation today at scale, we would be doing that. So very interested in learning about advances in that particular area. We're going to bring a slightly different element into this. We hope to be able to use a technology we have, a bioinformatics technology we have working in the lab, to try and identify cryptic vulnerabilities in these diseases. And this relies on the use of a tool called the Cancer Dependency Map, or DEP map. I didn't talk about it today, but maybe later. And then Dr. Wong has managed to find some collaborators in Texas who have 15 established PDX models of KMT2A rearranged pediatric AML. And we see some opportunity there for validation. This is another big problem in the field. How do you, what models are best suited for this kind of thing? They exist. And so here are some. So with that, I'll close. And so I'd like to specifically acknowledge Dr. Rachel Wong, who has been driving a lot of the more recent analyses and leading our thinking along a next series of experiments. Dr. Sander Lambo, who is now at the Dana-Farber Cancer Institute, for all of his contributions in our early experiments. And as Signe already shared with you, there's a view of our lab, which we call the Match Lab. Multi-omic analysis of treatment resistance and cancer heterogeneity. Come and visit us at the MSL. We're just setting up shop there now and pleased to be here. So thank you all very much. And thanks also to our funders, without whom none of this work could be done. And especially thank you very much to the families of cancer patients who participate and drive the research that we do. Thank you.
Wonderful. Well, thank you, Marco. Thank you, Marco, for that presentation and exploration of enhancer heterogeneity in the context of pediatric AML. I mean, as we wait for some questions to come in, reminder again that this needs to be done in the QA box, not the chat. I think this concept of enhancer or regulatory heterogeneity is something that seems to go beyond, obviously, pediatric AML, and we see this in other pathognomically described cancers, for example, synovial sarcoma or rhabdoid tumors. And it seems to be a feature that is associated with perhaps epigenetic dysfunction more generally, and I think is a real challenge for the field. And I think you're illustrating that. So maybe I can ask a question. KDM2A itself is obviously catalyzes H3K4 trimethylation. And I wonder to the degree that any of these, that our understanding of where that K4 tri is laid down in the context of these rearrangements is different, or there are any subgroups that emerge. Can you maybe speak to that? I guess this would have to be done in the bulk.
Yeah, I think it would have to be done in the bulk, and people have been working very, very hard to try and find these signals. And there have been experiments done on some of these things. I think the problem that we have and the problem that we face is that if we don't understand the processes that lead from a diagnosis to a relapse, if we consider only a single time point in this analysis, we lack the ability to observe the phenotypic change, diagnostic, relapse, and correlate that with changes in molecular properties. So, there have been lots of explorations of models. One that I came across recently was a paper from the Brian Wilhelm lab where they look at KMT2A fusions with MLL3. And so lots of these things have been done. I don't think that's, and when you look at them, they all sort of say, well, and this is the best that can be done right now. There's some dysregulation of the epigenome, and it's difficult to know what to make of it. And so I think the key here is we have to think about these paired samples in the next phase of things. And so I'm less interested as to where marks lie in diagnostic versus relapse. Or I'm more interested in that than I am where they are at diagnosis. Recognizing that some very large proportion of patients will respond quite positively to treatment. So the key to me is having those cohorts available, and those are really, really hard to find. So there's been stellar work done, Martin, on trying to work this out. And you can do some of these things. You can sort cells, so you can limit the heterogeneity if you have good markers for cell populations, and you can say, here are the marks. It's great. But how do they change with in this correlation to untreatable disease. I think that's the key, and I keep harping on this. And it turns out to be brutally difficult to get these cohorts, and we miss opportunities to profile them because, oh well, yes, of course, but we don't do it. So we have to do it.
Yeah, absolutely. And I think some of the work within the Terry Fox Research Institute Marathon of Hope, I think, is an example of where some of that work is getting done. I have a question that's come in from Christine, which is, what do you think makes KDM2A special in terms of frequency of rearrangement and in generating diversity compared to other mutated genes? So why KDM2A specifically in this context?
Yeah, so this is a good question. I'm not sure it's necessarily biochemically special, in the sense that there are other rearrangement types associated with disease. So it's not an exclusive thing. You have weird rearrangements of runks and this and that and the next thing, but it could very well be that KMT2A and other genes tend to be reasonably good sized, and they have lots of funny structures in them, which might make them prone to recombination events of a certain sort. And so when you think about the age of onset of these diseases, you might speculate that actually the disease doesn't start at birth. It starts in utero, perhaps, during development, where there's lots of cell division, lots of potential for copying the DNA content of cells, segregating to daughter cells again and again and again. Maybe KMT2A can be thought of as a somewhat error-prone gene that accumulates these things. There are other genes, and so we shouldn't lose sight of that. But maybe what it means is that when this happens, and when it encodes something that can dysregulate the epigenome and the transcriptome, it's very hard for normal biology to find and overcome. And so I think that's why we see these things. KMT2A is probably something that is a target of normal biology, or maybe only slightly dysregulated biology. It's big.
Thank you, Marco. So I think we have time for one more question. I know you have a hard stop here, and I think we're a few minutes over. So
we are.
I'm going to take one question, if that's okay. Could you? I think the question is just so this is from RT. Could you comment on targeting DOT1L in KDM2A rearranged leukemias and why it has failed?
It's the wrong target, and so I mean we're not aligning the way that we think about drug development. I mean, this is a very general comment. We're not necessarily aligning the way we think about drug development and testing with the biology of the thing that we're trying to inhibit. And the other thing that we have to be aware of is that when you have, I mean, this is my opinion, I think the literature would support it, but there's probably literature that would argue against it. But when you have something that acts at so many different sites in the genome and is capable of inducing huge amounts of cellular plasticity, and you throw a single target or a single inhibitor at that thing, I don't fundamentally understand how that thing is going to be overcome. Resistance will happen, definitely, because cancers are wildly dysregulated and highly plastic entities, and they'll work their way around targeted agents in general. So I think we have to be very careful about how we think about therapy going forward. I mean, there was all this delight and promise of immune checkpoint inhibitors, and that still remains. And we see in some patients that there's this possibility of cure. That's by activating a highly complex system that combats the highly complex cancer, your immune system. And even in that space, we run into this problem of relapse quite frequently, actually, and we still don't have perfect measures of aligning patients to immune checkpoint inhibition. It's more than just tumor mutation burden. So I really feel that with these kinds of diseases where they're driven by an epigenetic regulator that acts at, I don't know, how many million sites in the genome, where we look at things and we say, oh, well, things go up, things go down. Here's H3K27, here it's not. I mean, what's the target and what's the rationale for one target versus, something that is more systemic and more highly tuned to the phenotype of relapse. What is the phenotype of that cell? And can we imagine attacking that cell from a completely different angle? So that's not an answer. It's a plea for more research in this area.
Wonderful, wonderful. Thank you, Marco. And I agree that it is a very big question, and perhaps we can continue that discussion in Blue Mountain, where many of us will be in just over a week. And with that, I would like to once again thank both speakers for a fantastic seminar and a fantastic hour. And thank you all for attending. And with that, I will close today's seminar.
Thank you, everyone.